Good afternoon, everyone. Thank you for being here today. I would like to start by welcoming and thanking the members of the jury, Professor Anelisa Silva, the chairman of the committee, Dr. Megan Hedring-Roth, thank you for your presence, and my supervisor, Professor João Magalhães. My thesis name is the impact of 24-hour movement patterns on interstitial glycemia in people with type 2 diabetes exercising at different times of the day. It's a secondary analysis of the EPSAMI project. We'll first look at the epidemiology and the pathophysiology of type 2 diabetes, then we'll move on to the 24-hour movement behaviors and type 2 diabetes, then we'll look at the circadian rhythm and its impacts on exercise and glycemic control, and lastly we'll look at the original manuscript. We know that 589 million people today live with type 2 diabetes, and that number is expected to increase to 783 million people by the year of 2045. It has an economic impact of $966 billion, and the situation in Portugal is pretty similar, with 14.1% of the global presence living with type 2 diabetes, and it has an economic impact of 1.4 to 1.7 billion euros. To understand how insulin works, to understand how type 2 diabetes works, we first have to understand the insulin action. Insulin is a hormone responsible to lower our blood glucose, and it does that by binding into the insulin receptor and starting a bunch of cascades with phosphorylations leading to the translocation of GLUT4 into the cell membrane. It then captures the glucose and it brings inside the cell. We know that type 2 diabetes is mostly characterized by insulin resistance, and that insulin resistance cannot be attenuated with the increase of insulin secretion. We know that the number one risk factor for type 2 diabetes is obesity, and it is impacted by our genes and by environmental factors. We know that this is not a problem with the amount of insulin receptors, since with a loss of up to 90%, we still get a maximum insulin effect. Because most people have at least 10% of its receptors, we know that this is not a problem with the amount of receptors, but more of a problem with the translation signaling. And here we can see what happens in the adipose tissue. This tissue tends to hypertrophy with the increase of the calorie intake, creating a calorie surplus. And we see that it starts creating some necrosis, some hypoxia, which leads to the infiltration of M1 macrophages. These produce pro-inflammatory cytokines, such as IL-6 and TNF-alpha, and those lead to insulin resistance, leading to a lipolytic state of the adipose tissue, which affects not only the pancreas through the apoptosis and reduction of insulin production, but also the liver. Through these lipolytic states, the adipose tissue releases glycerol and free fatty acids. Those two get to the liver through the portal vein, and glycerol gets converted into glucose directly, and the free fatty acids go through beta oxidation and get converted to acetyl-CoA. Acetyl-CoA with pyruvate carboxylase leads to oxaloacetate, and that is the initial part of the process that we call gluconeogenesis. That's the formation of glucose, and to try to prevent the amount of increase of glucose, the liver tries to convert this into triglycerides through the process called de novo lipogenesis. That leads to the accumulation of lipid intermediates, such as acetylglycerol, which activates the protein kinase C, leading to insulin resistance. We see a similar behavior here in the muscle, where the free fatty acids get into the mitochondria. They start the production of the beta oxidation process, and that leads to the accumulation of long-chain acetyl-CoA, and that impairs the insulin pathway. We also get the formation of triglycerides, and that leads to the production of acetylglycerol and the formation of ceramides. These two activate the protein kinase C, which again leads to problems with the signal transduction. We know that exercise is a great ally to combat the problems with type 2 diabetes, and it does that by increasing the glucose delivery through the increase of blood flow. It also helps with the glucose uptake through insulin-independent pathways, such as the MPK and the CAMK, and it also improves our mitochondrial biogenesis through the increase of PGC1 alpha. These are the acute effects, and these here on the right are the chronic effects of exercise. But we know that exercise does not act equally to everyone. Some people respond really well to it, some people don't respond at all. And we can combat that by introducing a tailored protocol to affect equally everyone, and that's the process that we call precision exercise. We have some non-controllable variables such as the genetics and epigenetics. Some of them are partially controllable such as exercise adherence, and some of them are fully controllable, and today we're going to focus mostly on exercise timing. We know that one of the variables is the timing, and that is impacted by our circadian rhythm. It starts at the supraschematic nucleus, which gets information from our eyes, basically gets the information about the sunlight and activates the BAL1 and clock proteins. These two proteins then attach at the E-box, which is a specific part of the DNA, and that starts the activation of RNA polymerase to transcribe the cry and purr genes into cry and purr protein. These two proteins accumulate outside of the nucleus, and when there's enough accumulation, they then inhibit the activation of BAL1 and clock genes. At the same time that they're doing this, they also produce the ROR and reverb proteins, which attach at the ROR, which is a specific part of the DNA, and that makes sure that the cycle takes 24 hours. Not only does the supraschematic nucleus affect our peripheral clocks, but also the ingestion of food and exercise does. So with a modern lifestyle, we tend to give some mixed signals to our peripheral clocks, and that can lead to a misalignment of them, leading to an increased likelihood of developing type 2 diabetes. We see that some people, more than half of them, have a Down phenomenon. More than half of the people that have type 2 diabetes have Down phenomenon, and that is the rise of glucose in the morning period, because these people have a misalignment of the circadian rhythm, and that creates a more insulin resistance state in the morning period compared to what it's supposed to be, which is the afternoon. Looking at the observational literature, we see that this paper here done at the university, it has a sample size of 74 individuals, and it's a cross-sectional analysis of the different projects. It looked at MVPA while these people did structured exercise, and the participants used an accelerometer to track the physical activity. We saw physical activity from the time of waking up to 12 p.m. and they also saw the activity accumulated from 12 p.m. to 8 p.m. The results shows us that afternoon MPPA is more strongly associated with improved of HbA1c compared to the morning training. And when we look at the experimental literature, we see this study done by Rodrigo Manchila with 32 adults. It was a retrospective study of 12 weeks of both exercise, aerobic exercise and resistance type training, and it was done 3 times per week. We had a morning and the afternoon group, and the and the results showed us that there's more benefits in the afternoon exercise through an increased disposal of glucose, a better oxidation of carbohydrates, and a more free fatty acid suppression as well in the afternoon group. Looking at this paper from Slavic, this is one of the main papers from my thesis, and it was done with 11 men with type 2 diabetes. They trained, so this was a randomized crossover where they trained 2 blocks, 2 weeks of training each with a 2-week washout period, doing HIIT training 3 times per week. There was a morning block and an afternoon block, and as you can see here, the morning block has a lower area under the curve when we compare it to the pre-training. At the same time, we see that the morning training actually got a higher area under the curve when we compare it to the pre-training. But Michael Riddle, with the systematic review, tells us that there are still discrepancies in the scientific literature regarding the optimal timing of exercise, with some studies favoring morning and others favoring afternoon training. And this is where we get our literature gaps, which most studies just look at a small percentage of the day and exclude the other 23 hours of it. There's still some inconsistencies in the literature, with some studies favoring morning and some of them favoring afternoon, and we still don't know how much the structural exercise actually determined the sedentary behavior, and more specifically how different exercise timings affect that. Lastly, there's still not literature out there about the combination and the synergistic effects of both exercise and physical activity. And that's when we get my thesis purpose, which is to look at the 24-hour movement behaviors, exercise timing interactions on the glycemic control in people with type 2 diabetes. And my hypothesis is that all 24-hour behaviors interact with exercise timing to alter its impact. The inclusion criteria are adults from 55 to 75 years old with type 2 diabetes. They have to be medicated with metformin and SGLT2 inhibitors and GLP-1 agonists, and they have to be inactive with no more than 20 minutes per day, 3 times per week of exercise. Looking at the exclusion criteria, we see they couldn't be using any other anti-hyperglycemic medication such as insulin. They had to have no micro or macro complications with type 2 diabetes, and they had to have no positive criteria for the CPAT. And lastly, they could not have motor limitations that limited the practice of exercise and the inability to provide written consent. When we look at the study design, we see that this is a randomized crossover trial with 3 different blocks, 2 weeks long each, with a washout period in between. And we did the baseline and follow-up assessments. The baseline, we did DEXA scan, anthropometry tests, the CPAT, the physical activity assessment. We took blood samples, and we also recorded the glucose for 24 hours. And they follow up. We did the exact same thing, except for the CPAT. And there were 3 conditions: the morning, the afternoon, and the evening. And we did an adaptation phase before the intervention. During the intervention, we did 6 training sessions per block, 3 times per week, with bouts of a minute at 90% of the peak power output, and after that, a minute at 40 to 60% of the peak power output, with a total volume of 10 calories per kilogram per week. We did monitor the glucose through the use of a CGM from Abbott, the Freestyle Liver 2. It was a subcutaneous sensor located at the posterior part of the arm, and it took samples for 15 to 15 minutes for the whole 24 hours. We then got the mean glucose and the 22 hours area under the curves using the trapezoidal method calculated through the MATLAB software. This was done with controlled meals, which were 20% protein, 55% carbohydrates, and 22% lipids. The accelerometer we used was an ActiGraph GT9X in the non-dominant wrist, and to analyze that data we used the R software, more in particular the GGR package, and with that we used the validated cut points from Hillbrain. Looking at the methodology, the statistics part, we used the normality test using the Shapiro-Wilk test, and we got the mean and standard deviation as the descriptive statistics. To compare the baseline and the follow-up, we used the one-way ANOVA, and our main analysis was done using mixed models effects to see for 3-way interaction between time, exercise timing, and 24 hours behavior. The model was controlled for sex, age, and total wear time. We started with 52 participants. Only 27 of them enrolled in our study. 15 were allocated to the type 2 diabetes. We got 1 dropout, and 1 of the participants was excluded due to not meet the intensity criteria. When we look at the results, we see that we got 100% of the adherence. We also got just shy of 100% of the heart rate that we prescribed, and the energy expenditure was 83 to 86%, and the peak power and the VO2 peak was stable throughout all of the blocks. Here we see the descriptive statistics for each block and for each variable of the physical activity, and here we get our main analysis, which was with the mixed models effect. We see that throughout the sleep and the sedentary behavior, we don't see any statistical significance. In the LIPA, we actually see a statistical significance here in the evening period when we compare it to the morning and with the afternoon, and we see that with a positive beta. And in the MVPA group, or in the MVPA variable, we see a statistical significance when you compare the morning period with the afternoon period. And here we see a negative beta, meaning that the afternoon period got advantages compared to the morning one. When we plot this into a graph, we see here at the right, top right corner, we see that there's a steep curve here in the afternoon block, where we see that the mean glucose really gets lowered as we go through the exercise sessions, and that happens with while keeping the mean of the 24-hour movement behaviors, and as we kept exercising in the afternoon. Like I said, on the sleep, we saw no evidence that exercise timing alters the association between sleep and glucose control. The average participant got less than 7 hours of sleep, which is below the recommended amount of 7 to 9 hours, and that has a potential negative impact. It is important for future research to take into account the sleep quality, the sleep regularity, and the circadian rhythm synchronization. We see a similar pattern here at the sedentary behavior, where we did not see that the blood glucose responded to sedentary behavior was not impacted by the training schedules. The average participant got more than 8 hours, but we know that harmful effect of sedentary behavior can be mitigated through the use of exercise and MVPA, and that's why we only look at the training days, since we want to isolate the acute effects that exercise has on the sedentary behavior. We also did only use the training days, because we know that the movement behaviors could actually change if we got other days other than the days that people trained. Looking at the LIPA, instead of looking at it as like higher levels of LIPA in the evening or in the afternoon group were associated with an increase of interstitial glucose, we actually have to look at this with a little bit of nuance, and we see that we have a finite amount of time in a day, and if we want to get more LIPA, we'll probably have to misplace some MVPA. And that's why we conclude that the rise of glycemia is probably not a direct detrimental effect of LIPA itself, but rather a current reduction of the MVPA. And this is our main findings. We saw that during the afternoon training block, those who accumulated higher volumes of MVPA showed a significantly more favorable glycemic control, and we see that in observational evidence and interventional studies, we also see that there's superiority in exercising or doing physical activity in the afternoon, but here we have the first study that tried to look at the synergistic effect of both physical activity and exercise, and not just one at a time. We know that the Down phenomenon has a negative impact on the overall glucose control in people with type 2 diabetes, and because we saw better results at the afternoon, we can conclude that it might enhance the effects of MVPA due to a better synchronization of the circadian rhythm. We know that exercise helps by having an insulin independence pathways that leads to the GLUT4 translocation through the MPK and the calcium signaling. We also know that it helps with the transport of glucose through the increase of shear stress, which leads to the production of nitric oxide, and it also helps with insulin sensitivity through the depletion of glycogen, and that helps with the next 48 to 72 hours after the training session. But we got some new evidence that afternoon training might enhance the MVPA in glucose control by the synchronization of the circadian rhythm, leading to a better MVPA to glycemic control association. Our strength in this study was that we used a crossover design, so each participant was their own control. We also standardized the meals for the CGM. We monitored the physical activity using an accelerometer, and we controlled for confounding variables such as the weight, so there was no weight loss during the study. We have some limitations as well, such as short duration of the study, so each block only took 2 weeks to complete. We also did not control for the MVPA distribution, and this is something to take into account for future research, and we used a small amount of GP1 participants. Foi melhor essa?
Foi.
Achas?
Tu, mas mesmo que tu te engajes, não faz mal. Também se falasses português também era a mesma coisa.
Epá, estou fedido da garganta.
Pois estás.